Thank you, Tom. So it's my pleasure to be here today to discuss some of the work that I've been doing on specifically estimating mosquito abundance. The outline of my talk, first I'm going to give a little bit of a background about how people, some simple approaches to estimating abundance from count data. I'm going to give an even simpler overview of mathematical models of mosquito-borne pathogens and let you know where mosquito abundance does or more often than not does not fit into those models. And then finally, I'm going to go through two case studies applying the methods I'm going to describe. The first being modeling the Aedes aegypti abundance in Iquitos, Peru, and the second modeling some Culex and Aedes species within California. So typically, when people want to model count data, the first thing that they can think of doing is using a Poisson regression. So under very simple assumptions, count data can be modeled using a Poisson distribution. Poisson distribution has the property that the mean is supposed to equal the variance, and very typically, when people actually go to fitting data, they find that the variance is quite a bit larger than the mean. This is called over-dispersion, which means that maybe a Poisson distribution isn't quite the right thing to be doing. Some people then go to a negative binomial, but what really might be happening is that some of the variance that we are observing in this count data might be being explained by covariates. So the way that one would go about explaining the relationship between covariates and count data is through Poisson regression models. For example, you might think that the colder the temperature, the fewer whatever you're looking at might be present. So you might try to have a relationship between temperature and abundance, and then when you incorporate this covariate, you'll actually see that that extra variance that you weren't explaining before is explained, and then it actually does follow a Poisson distribution with that covariate in mind. So I'm going to be presenting some work on a particular type of Poisson regression, specifically one that uses generalized additive models. So a generalized additive model is a type of regression model. For example, I see here we have the day of the year and we have temperature. And if I wanted to fit a regression model to this, I might try to figure out, okay, it doesn't look very linear, it doesn't look very quadratical, maybe it's, because it's periodic, maybe I should try fitting some sort of sine function to it. Generalized additive models don't force you to pick a particular functional form for your relationship. However, what they try to do is they try to fit a smooth function through the data. So for example, this would be the smooth function fit through the data. I'm going to continually throughout the talk be using the GAMS as implemented through the MGCV package in R by Simon Wood. And for those familiar with regression in R, it's actually really easy to use GAMS in R. This is actually all the syntax required to fit a relationship between temperature and some arbitrary function of day. So where does this all fit into mathematical models? The basic mathematical model framework that you can think of for a mosquito-borne disease is as follows. There's a couple tweaks depending on what pathogen you're talking about, but typically we have susceptible humans being infected by infectious mosquitoes. They go through an incubation period. They themselves become infectious, and when a susceptible mosquito bites them, then the mosquito goes through the same sort of pattern. Here I actually diagrammed something like dengue, something like malaria. The humans would go from the recovered class back to the susceptibles. So there's been a lot of work developing different tweaks on this type of model, making them more complicated. Something like West Nile virus is much worse with multiple hosts and multiple mosquitoes all interacting in complex ways. But recently we did what we're interested here is looking at the actual abundance of mosquitoes and how that affects what goes on with the mosquito part of the diagram. Recently we did a systematic review of all mechanistic models of mosquito-borne pathogens. Of the 388 mechanistic models of mosquito-borne pathogens, incredibly only 4 allowed for survival rates of mosquitoes to be spatially dependent. Only 17 considered the effective temperature in any way whatsoever, whether it be realistically forcing using temperature or just using temperature in a very silly non-database way. And only 7 of the 388 mechanistic models of mosquito-borne pathogens used modeled mosquito density using data. Now, it's not to say that people don't model mosquito or other insect density using appropriate population biology tools. It's to say that these tools have never, 7 times, have been incorporated back into a mathematical model of mosquito-borne pathogens. So one of the things that one of the goals of my work, or this part of my work, was to try to develop products that would take mosquito density data, convert it into something that we can then stick into a mathematical model, and then we can test things about the importance of accounting for actual true mosquito density. At the end, hopefully I'll convince you that that's a worthwhile goal. So let me talk first about the first case study. This is work that's been ongoing with many different people, most notably Steve Stoddard, Amy Morrison, and Tom Scott from UC Davis. And we're trying to model estimate the abundance of Aedes aegypti and also test what the effective control is in Iquitos, Peru. Iquitos is a city of 400,000 people. It's spatially isolated from the rest of Peru. It's at the beginning of the Amazon River. Dengue virus has been there continually since 1990, and the Scott Lab has had a presence there since 1999, collecting data on both human hosts and mosquito vectors. The mosquito vector of dengue, the primary mosquito vector of dengue is Aedes aegypti. It's a peri-urban mosquito, primarily living in and around homes. There's no problem living in a city. And it also it's not a very long distance flyer. So here its dispersal is typically limited from 40 to 100 meters. Some recent studies are actually showing that most the mosquitoes don't disperse past 10 meters. So it doesn't move around very much. Dengue, if you've heard a talk by Tom before, dengue is a arbovirus, the most important arbovirus in the world, causing approximately 200,000 deaths a year from dengue hemorrhagic fever. It is caused by 4 related but antigenically distinct virus serotypes. So what we're trying to do here is understand the role that mosquito density is playing in the ongoing transmission of dengue within Iquitos. The data we have is household aspirator data. So someone will go in essentially with a vacuum cleaner into a house, vacuum up all the corners of the house, and then count the number of mosquitoes that they've actually collected. Well, they'll determine the species of all the mosquitoes, and then they'll count up all the ones that we care about. So here we have 194,046 different household aspirations. The dots are very small. When they clump up, that means there was a lot of observations at the same place. And in these 194,000 collections, only 97,000 mosquitoes were caught. I mean, it's easy for me to say only, I didn't have to count them. But if you think about it, that means on average we're collecting about half a mosquito every time we go out. There's not very many mosquitoes. At least there are not very many Aedes in Iquitos. If you look at the same data temporally, so in this top panel I have the number of collections per month. So on average you can see about 1,000 household level aspirations per month. And if you squint your eyes and you kind of want to believe it's true, you might be able to see some sort of seasonal pattern. Maybe in the beginning it kind of deteriorates here, and maybe there's some sort of seasonal pattern later on. One thing I need to make sure I note, this red dashed line, we upgraded the vacuum cleaners that we went into the city, into the houses with. Independent tests estimated the new aspirators collect about twice as many mosquitoes. I'm not going to talk much more about that, but I'm incorporating that into the modeling approach. Now, dengue, like I said, has been in Iquitos since 1990, which means it's been infecting people since 1990. It's been a public health concern since 1990, so not surprisingly, the government has been going in and doing their own control efforts. So here we have 6 different citywide control efforts. They don't actually cover the entire city. They don't actually cover entire zones. But what maybe again, if you squint and want to believe it's true, you can see maybe when you have a red arrow indicating control, mosquito population dipped. Sometimes it dips, sometimes it doesn't. So what we'd like to really do is try to understand what exactly is the level of variation in the seasonal variation of the mosquitoes, the year-to-year variation of mosquitoes. We also like to see if there's a spatial differentiation in where some places there are a lot of mosquitoes, some places there aren't very many mosquitoes at all. And then again, for the purposes of then connecting this back to a mathematical model of dengue, we want to have a predictive tool or a predictive product that if you give me a location in the city and a time of year and a particular year, I can tell you how many mosquitoes you expect to see there. So we can then see if that matches up with incidence data. And then finally, we have this control data. It's a little unclear exactly what role the control data is playing, because maybe sometimes we're doing control where there aren't very many mosquitoes in the first place. So we want to be able to see if there's a way to evaluate the effect of the control efforts, and if appropriate, incorporate that back into the product. One of the reasons we'd actually care about estimating mosquito abundance, if it actually has a huge role in transmission dynamics, we'd like to be able to then poke a model that incorporates mosquito density with things like what would happen if I sprayed here, or what would happen if I sprayed there. So we want to actually know, well, what is the effect of spraying somewhere? Because spraying, like everything, won't be perfect. So we want to evaluate how imperfect control efforts are. Results. So I'm going to skip actually telling you how I did most of this. It is actually not very difficult, so please come talk to me later if you'd like to know. But we can see here the seasonality and the interannual variation of mosquito abundance. So here we have 2002, 2001 was a bad year, 2004, 2005 was a good year if you're thinking about not having many mosquitoes. And then we also have some sort of interannual variation. Right around November seems to be the height of mosquito abundance, and right around May seems to be the trough. One of the things I'd like to bring your attention is the y-axis here. This is 1, meaning we expect 1 mosquito every time we go out. This variation, while significant, we have 194,000 observations, so we can draw some pretty significant, statistically significant differences. This variation isn't really that large, from the biggest year to the smallest year, or from the shortest part of the year to the largest part of the year. So the variation, while significant, isn't really that, doesn't seem like maybe it has that big of a practical importance. If we look at it spatially, we see the same sorts of things. Here we have places, sorry for not having a y-axis or z-axis on this. Here we have places within the city that systematically underproduce. So these places have about 1/10 of a mosquito every time you go out. And then we have other places in the city, like here we have 1 1/4 mosquitoes every time you go out. The modeling approach not only estimates the mean abundances, it also estimates uncertainty. So this bit up here, if you remember from a couple plots ago, which you probably don't, there's not much observations up here, and the very few times they went out here, they collected a lot of mosquitoes. So while the estimated mean is quite high, the variance is quite high as well. Whereas places like this, which is actually a highly populated area, has a relatively high mosquito abundance, but the certainty is much smaller. So then, again, we want to think about the effective control on this data. And again, these aren't varying very much from about 1 1/4 mosquitoes to about 1/10 of a mosquito. So if we think about going to a location in the city 2 years in a row, and we compensate for the amount of mosquitoes we think are going to be in 2008, for example, versus 2009, we're going to the same spot, so we think we'll probably get a little bit fewer mosquitoes, but we know that there was control in that location. We'll then see, we hope we see fewer mosquitoes than we expect, and we can try to model the relationship between the amount of control we have in place and the decrease in the abundance of mosquitoes. We get the following plot. So again, we only have 6 really large citywide spraying control efforts that didn't actually cover the whole city, but within those, we can estimate if you have, for example, in the absolute middle of the city in 2010, with no control, we estimate about almost 7/10 of a mosquito every time you go out. And if you were able to completely spray the neighborhood around that house, then we'd estimate about 2/10 of a mosquito coming in. So again, these are really small numbers, and so what you might be asking yourself, does any of this actually matter with relation to transmission to dengue? I have significant effects, but that's could be just because I have so much data. In reality, it's hard to actually link mosquito abundance directly to transmission. However, we can see there seems to be somewhat of a strong effect of mosquito spraying. If we try to correlate spraying directly with dengue incidence, we see a relatively striking pattern that does suggest, well, not surprisingly, given that it's a mosquito-borne pathogen, does suggest that mosquito abundance matters quite a bit. So this is going to take a little bit of explanation. In this left panel, we have the first half of the transmission season, so about September to January. The dots here represent the number of cases of dengue observed in 1 week, and then the number, that's the x-axis, and then the y-axis is the number of cases observed of dengue in the 3 following weeks. So what you'd expect if transmission is holding constant, you'd expect about a slope of 1/3. So that's this red dashed line here. If you regress the black dots, these black dots represent weeks where there was no spray effort. You see that they actually pretty much perfectly follow this, everything's going pretty much constant. If you regress the green dots, and a green dot represents a week where we sprayed, and then we looked at what happened the following 3 weeks, we see a significant decrease in the amount of dengue in the following 3 weeks. So even though this abundance is quite small, spraying mosquitoes, which is reducing this really small number to a smaller number, seems to be having quite a large effect on incidence. Yes?
Were the sprayings randomly throughout the season? Happen at the same time of year every time?
Neither. They would typically be in response to a large outbreak. And so that wouldn't always occur at the same time of the year, and they would try to not have it be completely random in the locations they would spray. but they wouldn't spread perfectly throughout the city. So as a first, integrating across a lot of variation, there's still this strong effect of spraying unobserved mosquito abundance to dengue. However, if you look at the second half of the transmission cycle, so this is from about January to about May, or maybe March, you see, again, the same plot, same setup, cases 1 week, cases following 3 weeks. Here's this red dashed dotted line showing what we'd expect if it's constant. But again, this is the end of the transmission cycle, so things should be going down. So we'd expect things to be a little bit lower, the slopes to be a bit lower. And maybe you can't quite see, I can see from here, the black dashed line and the green dashed line are right on top of each other. So they're both on their way down, and spraying doesn't seem to matter. So in the first half of the year, spraying, which then I would argue translates to mosquito abundance, makes a difference in what's going on with dengue incidence. However, I'd argue in the second half of the year, spraying, which will affect mosquito abundance, isn't necessarily having such a strong role in dengue incidence. So mosquito abundance matters, but it's not the whole story. So in summary of these results, there is significant seasonal and interannual fluctuations in abundance. The locations within the Iquitos, some locations within the Iquitos systematically over and underproduce mosquitoes. Control, when applied throughout an entire region, appears to have mosquito abundance. And further, although Aedes aegypti aren't actually very plentiful in Iquitos, there is in my mind, clear connection between spraying and dengue outbreaks. However, this connection is temporarily differentiated, meaning that mosquito abundance and accounting for mosquito abundance alone isn't going to be able to give us all our transmission dynamics. Moving on, second case study. So this is work with Bill Risen and Chris Barker in California. Again, I get to be lucky and I show up at the end when all the data gets to be analyzed and I don't actually have to collect the data. We're going to be talking about the seasonality, temperature dependence, and land cover dependence on abundance of arboviral mosquito vectors. So the mosquito vectors we're going to be talking about are Culex tarsalis, the Culex pipiens complex, and Aedes melanomen. The pipiens complex and Aedes melanomen are both vectors for West Nile virus. Culex tarsalis is a vector for several encephalitis. I'm not sure that's the right way to pluralize that. And both the Culex and Aedes species are potential vectors for Rift Valley fever, which isn't actually in America, but were to show up in, for example, the Central Valley of California, would have devastating effects on our cattle industry. All 3 or 4, depending on if you want to combine pipiens with Quinquina fasciitis, are present in California and quite abundant. West Nile virus, this is probably something people are quite familiar with. Birds are the primary host of West Nile virus. Humans, while being able to be infected, are dead end hosts. The first West Nile case was reported in America in New York in 1999. Spread across the country 3 years later, the first case was reported in California. One of the reasons probably people know about it is in 2012 it killed 286 people, the majority of them in Texas. California trap data. So this plot is set up pretty much similarly to the last one. In the last example I gave you, though, we were talking about aspirating data, or aspirating counts. So I'd go into a house with a vacuum and suck up all the mosquitoes I could find. Here we have a different type of data collection. We have CO2 traps. And so CO2 traps were set out, pretty much scattered across California from 2003 to 2009, and there were 102,000 trap nights. Again, the mosquitoes I'm talking about in this example are quite a bit more abundant than Aedes aegypti. In those 102,000 trap nights, 4,882,911 mosquitoes were caught. I feel I have to go through that number specifically because somebody then went through the effort of determining the species of every single one of those, which, again, I'm quite happy I didn't have to do. If you look at the data in time, as opposed to in space, you can see here on the top panel we have the effort, the number of trap nights per month. You can see the number of trap nights per month has increased as the years have gone on. There's still a seasonality to it. And as well as there being a seasonality to the actual number of trap nights, there's a seasonality to the abundance. The first couple of years you can see are a bit higher, but then from that point on it seems kind of constant. One of the things that I'm not going to be asking with this data is if there's a lot of interannual variation. But there is a lot of, in this case it's a lot clearer that there's intra-annual variation or seasonality, and so we're going to try to figure out what the effect of seasonality is. For every trap night, in addition to the number of mosquitoes that were caught, we have the temperature of that trap, or at that trap, and the land type for the surrounding 50 meters. And obviously, if you go 50 meters out, you're not going to have one type of land type, so we have a breakdown of 50% grasslands and 50% pasture, for example, for a particular place. What we're going to try to do is figure out what the effect of seasonality is, what the effect of temperature is, and what the effect of land type is. So let's consider 3 different locations within California. And this data is a bit of a toy data example. So it's not to say that if you went to the Central Valley, you'd find this exact breakdown of land type. But let's just say you did. So if you went to the Central Valley and you found the majority of the land type was pasture, a little bit of scrub, and a little bit of urban, maybe you'd expect throughout throughout a year, there's not very many mosquitoes, and then at some point in time, the mosquito abundance peaks. Maybe if you went down to the Inland Empire in Southern California, where it's primarily urban, you'd expect to see somewhat of a similar pattern, but it's primarily urban, so we're not going to have as many mosquitoes. Maybe if you went out here, you'd expect to see mostly scrub, and you'd have even fewer mosquitoes. Again, this isn't actually a true representation of out here, but this is just to show what maybe one would expect. I'm going to specifically try to explain the problem, the objective of my data, by thinking about this one location and its breakdown here. And basically what I'm trying to do is I'm trying to use the data as an inverse problem to take this signature in time with this known breakdown and split it into its constituent parts. So I have the data aggregated, or I have the data when you look at a particular composition of land types, and I want to figure out what part of that temporal signature is due to shrub, what part of that temporal signature is due to urban areas, and what part is due to pasture. And so maybe in this particular example, the pasture has quite a few mosquitoes. You can't see the axes here, I can. It's about 40 mosquitoes a night. That's quite a lot. Whereas the shrub area has only 2 or 3 mosquitoes a night. But when you take the weighted average of these 3 land types within that one location, you'd expect to see the aggregated temporal signature. So to skip right ahead to the modeling approach, what we're trying to do is we're trying to figure out, is there a interaction between land type and seasonality? And specifically for a given location, if we break down that location into its constituent land types, can we figure out what the signature for each land type is seasonally? As well as, is there a relationship between temperature and abundance, in addition to the seasonality relationship? So obviously, during the summer it's hotter, during the winter it's colder. So there's going to be a natural relationship between seasonality in a very vaguely defined way and temperature, but I want to see, in addition to accounting for seasonality, is there still an effect? If it's June 1st and it's a hot June 1st, is that going to give me more or less mosquitoes than if it's June 1st and it's a cold June 1st? The modeling approach specifically, again, this is the only really dense math slide I think I have. What we're going to be trying to do is we're going to try to have the log mosquito abundance is going to be a function of the seasonality with relation to land type times the proportion. So for a given location, I have 50% urban, 50% pasture. So in that particular place, I'm going to try to say the mosquito abundance there is 50%, 0.5 times whatever the seasonality of pasture is, plus 0.5 times whatever the seasonality of urban is, plus some relationship between temperature. Again, site-specific temperatures. This model is known as a varying coefficient model. It's kind of a little funky model because I'm fitting S, these 2 guys, but I'm not fitting the P's because they're specified. The reason I'd want to do this is that then if I have my locations, I can extrapolate from my locations, but for anywhere else in the country, or the state, maybe the country, but anywhere else in the state, I can say what is the breakdown of land type there, what is the temperature at a given point in time there, and then by recombining these seasonal signals for the different land types for each individual location, I can create this abundance product that I was talking about that I want for mathematical models of infectious diseases for any location within the city, within the state, based on the data we've collected. So again, skipping the details, I'm going to jump right to the results. So we have 9 different types of land type: water, urban, barren, forest, scrub, grass, pasture, crops, and wetlands. I've displayed the resulting seasonal signatures on a log scale. So for example, water. If I was at a place that was 100% water, which wouldn't really be possible because the CO2 trap would have sunk, but if I was at a place that was 100% water, I'd expect to see about 2 mosquitoes every trap night. And there isn't much of a relationship between seasonality and water land type. However, on the other hand, if I go to someplace like the wetlands, I see quite a significant dip in mosquito abundance, and then later on I see a jump in mosquito abundance. Some of these land types correspond to places where people are systematically changing the larval habitat. So you might imagine crops might be getting water more frequently, or particular places might be getting flooded for different agricultural applications. And so what I'm really incorporating here is I'm incorporating not just the natural larval habitats as they change through time, but also what man is doing to these things. Finally, this last one, I have my 9 land types, and I also have temperature. So we go from -10 Celsius up to 40 Celsius. We see a significant increase in mosquito abundance up to about 30 degrees Celsius, and then a significant decrease in mosquito abundance because the mosquitoes are basically frying at that point. If we had data, we wouldn't be surprised if it was 50 degrees outside, all mosquitoes die instantaneously. So what can we do with this now? So for example, we can ask the question, if we went to a place that was half barren and half scrub, how many mosquitoes would we expect to see? So in this particular case, barren and scrub are two places that don't have very many mosquitoes on average. So if we went at the peak time of year, we expect to see 2.5 mosquitoes. If we were to think then, what would happen if we did some sort of, if this barren scrub location was on the edge of Los Angeles County and the urban sprawl slowly took over, how would we expect the mosquito abundance to change if we took 1/3 of this area and made it urban? Well, we can do that very easily with the model. We can see that in fact, if we increased the urban percent of the land cover, we'd go from about 2.5 mosquitoes to 3.5 mosquitoes at the peak. So in this case, urbanization would be bad. On the other hand, if we went to some place that was half grass, half pasture, both of these types of land type actually produce a reasonable number of mosquitoes. And if we were to take this and urbanize 1/3 of the area, we actually would decrease the percent of mosquitoes we'd see. Finally, and this is not necessarily the best application of this, but we can ask ourselves what would happen if we had global warming. So we have our temperature relationship, and if we considered that one location I was talking about before, there was a reasonable percentage pasture and a small percentage shrub and urban, and we asked, okay, what would we expect to see on the average July temperature? Okay, this is how many mosquitoes we'd expect to see. If we then increase the temperature by 5 degrees, we would see mosquito abundance increase. One thing I'd like to point out, the increase in some places is quite large, but an increase in some other places is quite small. And the reason for that is that these times of the year are already naturally warm. So if we're increasing the temperature, we're actually getting closer to being on the downslope of the relationship between temperature and abundance. And obviously there'd be a lot worse problems than mosquito abundance if we increase temperature by 10 degrees. You can actually see there's starting to be a decrease in the mosquito abundance, again, because now we're starting to fry the mosquitoes. So the summary of these results. There exists strongly significant interaction between land type and seasonality of mosquito abundance. Hopefully those plots convince you of that. The p-values are tiny, and again, as opposed to with the Aedes aegypti, these are actually quite a significant number of mosquitoes, different from one point in time and another point in time, from one land type to another land type. The relationships don't appear to follow any particular functional form. Again, you might think, oh, these are periodic, so maybe I should do some Fourier analysis on it. Sinusoidal functions don't really seem to be appropriate, and this flexible tool of generalized additive models seems to be appropriate and seems to give us what we're looking for. There appears to be a nonlinear relationship between temperature and mosquito abundance. We are having, not surprisingly, there's a temperature range where mosquitoes are happy. If it's too cold or too warm, they either freeze or they fry. And again, the final goal for me is to produce a product that I can then put into a mathematical model of, in this case, West Nile virus, or perhaps a predictive model of Rift Valley fever, and say, across the state, where do we expect the most mosquitoes? Where do we expect fluctuations in mosquitoes to be the largest? And we can ask questions about how important that is for transmission dynamics. So then actually, finally, in conclusion, mosquito abundance is a key component to mosquito-borne pathogen models, and it varies considerably in space and time. Hopefully, at least that one slide showing you the relationship between spraying and dengue incidents convinces you that mosquito abundance matters. Unfortunately, as I mentioned earlier, only 7 mathematical mechanistic models of vector-borne diseases actually cared to go through the effort of incorporating real-world data into their mosquito abundance, so one would ask how useful those models actually were. Modeling spatio-temporal pathogen transmission, as well as modeling localized control, seem to require some amount of understanding of spatially and temporally dependent of mosquito abundance estimates, And so hopefully by producing spatially and temporally dependent mosquito abundance estimates, we can start answering questions in a much more accurate and systematic way. As I mentioned earlier, there are strong relationships going on here. Sometimes they don't actually appear very practically important, but they're strong relationships and they don't seem to follow a functional form, suggesting GAMs are appropriate. Generalized additive models provide a flexible tool when they're incorporated with Poisson regression for modeling abundance data. If you went through the effort of doing this and you still found you had overdispersion, there's absolutely no reason why you couldn't incorporate a GAM with a negative binomial regression. Any generalized linear model you can think of, you can then stick GAMs onto it to allow for flexible functional forms. And then finally, GAMs used in conjunction with Poisson regression can be applied to any abundance data set, not just ones for mosquitoes. It does help if you have a ton of data when there's a weak signal. And that's one of the things, if you don't have very much data but there's a strong signal, it, like everything else, should be able to identify it. But if there's a weak signal but it's there, it's kind of hard to actually tease that out unless you have something that's flexible like GAMs. And with that, I'd like to thank you and ask if there are any questions.
How did you account for species difference in land cover production in the California data?
So the results that I showed were just for Tarsalis.
Oh, okay.
But I have the same results for the Pippin's
so
complex.
it's different by species.
Yeah. And there's a significant difference between at least the A's melanomen and the Tarsalis.
Sure. But I don't remember you saying that.
Yeah, sorry. Yes.
So two questions. One that's very narrow and one that's more broad brush.
Cool.
So the narrow one, on the dengue control, I think if you had it as a first trimester, is that what you said, the left panel?
Sure. Yeah.
If you go back there. Okay, there we go. The post-spray or treatment, whatever it was, regression there, obviously has a lower slope, but it also has a higher y-intercept. And so if I look at sort of that cloud of green points and I sort of compare it with the cloud of black points, it doesn't, I'm sort of, yeah. Is that a real good, is the slope what we're interested in, or what are they interested in?
So I think the first thing I'd point out is that there aren't any green points close to the actual intercept, because they wouldn't have gone out to spray if there weren't very many cases. So using a linear relationship, the whole talk I'm arguing about using flexional relationships, and then I show this using a linear relationship, which is probably not a good thing to do. But using a linear relationship, it's not really appropriate to, in this particular context, to think about what this intercept means, because you're extrapolating well past the boundary of where these green dots show up. So, I mean, basically you don't have a green dot past this point here. If I fit some nonlinear relationship, or if I fit a GAM to this, I'd imagine the thing would, well, I imagine the thing would trough a little bit, but then the variance would become gigantic because I don't have any data out there. But that's a natural, that's the natural reality of the world, because no one's going to go out and spray if there aren't very many dengue cases. I mean, it's a good observation, but I think in this context, it's not a problem. It'd be weird if it was.
Okay, so let me ask the more general question, which connects with this, I think. So let me first say that I'm totally sympathetic to like I mean, I like a lot of the analyses you presented. I've been doing remotely similar things, and the challenge that I get from reviewers, and I'm wondering how you deal, if you get the same challenge and how you deal with it, is the response is these are all correlative data sets. How do you infer causal relationships based on these correlations? And I think that this data set is a good example. So to say, well, they only spray if the densities are high, so then those two populations of data points are not really directly comparable, right? There's a non-random selection of sites that are targeted for spraying. So how do you get that response? And if so, how do you cope with that?
So I guess the easier thing to do is that typically we're just trying to point out correlative things and not and draw a suggestive causal relationship with this. This plot is actually not from the same analysis as the mosquito abundance data, and one would hope this is just literally summarizing the reality of Iquitos and dengue incidents. And so it is what it is. It may or may not actually be suggesting something. I think it's suggesting something pretty strong, but you're right, it is absolutely non-random, the decision to spray. And again, it's not really clear that when they go out to the city, they actually know what they're doing with where to spray. So they could be spraying in the wrong place where dengue was 2 weeks ago or where dengue never has been. And so it's kind of hard to judge. Hopefully, given this effect that mosquito, so this is based purely on the data. Hopefully, given this effect, when we then take our spatio-temporally explicit mosquito abundance and stick it into a mathematical model of dengue that can allow for spatio-temporally explicit mosquito abundance, and then poke it, we can see that the dengue incidence data, which we also have spatially and temporally explicitly, looks at something more that suggests something a little bit more positive. It isn't very easy to ever draw causal inference from anything ever, but I think in this case, this is exceptionally suggestive, and this is one step closer to us being able to make an actual claim that spraying decreases incidence. Sometimes, right? I mean, this also suggests that spraying sometimes is useless. So it's all very complicated and founded. Yeah.
Is there any validation done on the predictions for mosquito abundance by land cover and temperature?
What do you mean by validation?
Well, so you're predicting that under different circumstances there would be a set number of mosquitoes. Did you validate that either with data that was already collected, or is it being prospectively validated?
So the way that GAMs fit a model, they actually try to minimize the cross-validation error score. So what they do is they drop a bit of the data, they fit a smooth function to the rest of the data, and then they try to see how well the model would predict the data that they left out of the model. So kind of inherently within it, they're doing some amount of validation. Beyond that, the residual analysis that we've done so far suggests that there isn't large patterns that we're missing, at least against the covariates we're not including in the model. There is some amount of a three-way interaction between seasonality, land type, and temperature. But of course, when you get to three-way interactions, you start having a huge number of parameters. And so while there is a sort of suggestion of this signal there, it's not large enough to be statistically significant. There's a different effect in temperature depending on which land type you are, depending on which type of year you are, but it's not statistically significant. So there's a little bit of a residual, you can see a little bit of a residual effect there. Yeah?
So if you could go to one of your last sort of climate change slides with the some of those curves, if you warm things up.
Yeah.
So I think actually I wanted to see maybe the next one.
That's the last one.
Was it? Okay. Right. So one thing about this, I wonder if at some point you might get into trouble if you don't consider mechanisms. So for instance, here it sort of looks like, well, if things warm up, it almost looks like mosquitoes sort of wait until like the right time of year to sort of pop up. But in reality, if they sort of get knocked out early, they might all die, and then you wouldn't actually have this sort of rebound that you get based on this prediction. So I don't know, could you sort of comment on that?
So yeah, the use of extrapolation, and again, I mean, I like this slide because you can ask the question, what happens if it's a little bit colder this year than last year? Or what happens if it's a cold front and it's the warm temperatures wait a little while? But typically when you're asking these questions, you're talking about a degree or two. When people talk about global warming, they typically just add 5 degrees to things and they go kind of crazy. In addition to wanting to actually understand what the mechanism for mosquito abundance would be, it's a little inappropriate to think that mosquitoes wouldn't adapt or move or have some sort of, perhaps mosquitoes that are more able to deal with warmer temperatures would move into this region. There are certainly, and again, there are certainly different locations within California, same land type, same species, some mosquitoes can handle hotter temperatures than others, depending on where they are, just because they're used to it being hot, so it's not so bad. And again, I'm not really able to take that into consideration with this, but it is definitely something. If you wanted to add 10 degrees Celsius to things, and you had a really good reason why to do that, you definitely wouldn't want to take this with anything other than a giant grain of salt. Yes?
Kind of to extend that question, these curves are just what you'd expect the core of the data to be, but they don't incorporate mosquito life in the street or anything.
Right.
So you're basically saying, like, if there was a knockdown, generation time effect, blah, blah, blah.
Yeah.
So how would you go about incorporating, in general, life in the street facts into a population model like this?
So there are other relationships. So this relationship is quite close. This takes forever to load. this relationship is quite close to an empirically observed relationship between mosquito death rates and temperature. And so as a mosquito has on average mosquitoes live 12 days. If it's a little bit warmer, they die a little bit faster. If they live a little bit colder, they die a little bit faster. And so the way we're actually using this product in a West Nile virus model is to use it for the number of mosquitoes that emerge at a certain point in time. But then we can actually ask the question, well, first of all, given whatever temperature it is, on top of knowing that they emerge at a certain point in time, we can say, okay, well, how likely do we think that they're going to die? Because obviously if you have 1 million mosquitoes, but they're all 4 days old, and they always die after 4 days, no one really cares, because the incubation period of all these pathogens is longer than that. So this bit only really provides an understanding of how mosquitoes are emerging under situations close to those where the data is collected. If we try to extrapolate too much, we really want to start thinking a little bit harder about how we're actually going to translate these into something that's useful for a vector-borne disease. Yes, Bill?
Just to add, of course, this doesn't account for water and breeding sources. And of course, when it's hot, in the middle of the summer, it never rains in California. So during that period, a lot of the natural sites dry out and they're no longer producing mosquitoes. So it's not always intuitively, it looks intuitively that it relates to temperature tolerance, but in fact, it probably relates as well to the availability of production in the general demography and population. And the areas that don't show the big decline areas are areas that they're irrigating, for example, like pastures. And the blips in the fall are due to flooding up of marshes and other anthropogenic effects with water. So it's Bobby's working really, really hard to make something functional out of a mess. Statistically, to do all this.
So a lot of this data is from United States. You're trying to do it for the entire United States area, or it could be applied to other countries. Because, like he was saying, summer here is dry, but summer Southeast Asia is wet. So I think it would be interesting to see maybe how likely mosquitoes affected. You can see them in different countries coming here, which countries are most likely to send them to us, or
yeah, so the kind of the state of the art that people do right now for trying to estimate malaria distributions, they look at the climate suitability of the mosquito, and they actually look at much weaker relationships than we're having. They basically just go crazy with this temperature abundance, this temperature relationship, and say, oh, mosquitoes wouldn't be able to live long enough, and they wouldn't have the extrinsic incubation period of malaria wouldn't be long enough. So the data we have on one hand is a lot more detailed and theoretically could be then applied to other places. But again, it's really hard to know what the anthropogenic influences are and how to tease that apart from what we're seeing. So if we go somewhere that has 100% barren, but it's in the middle of some other country, who knows what effects that we're just kind of integrating over versus what they're doing. And we certainly could, and I think one of the next steps for us would be to try to do it just for the whole country, because we have California and we have land types and estimates of temperature, daily temperature for the rest of the country, and so we could try to project over and see what we think would be going on. But again, not throughout the whole country, even if a place is suitable for a particular mosquito, it doesn't mean that that mosquito exists there.
Do you know how they come over? Do they go over on ships, or how
how the mosquitoes?
Yeah, do mosquitoes come, or just someone infected with the disease?
Oh, I think the mosquito. Well, for West Nile virus, the birds come.
The birds, yeah.
Yeah.
But remember, this is for one species, so the other 3 species that he mentioned have different patterns. And if you were interested in Japanese encephalitis in Asia, where you have a big summer monsoon and rice production, you'll get totally different patterns because of the land cover changes, like Bobby mentioned. So mosquito's not a mosquito, and each of them are really different, and these curves all vary. That was some of the madness that Bobby had to deal with. Our 80s are very different, for example, the curves.
Yeah.
Okay, thank you, Bobby.